All right, guys. Last but not least, the genomics session. Good afternoon. My name is Helen Lu. I'm a faculty member in biomedical engineering, and my area of expertise is not in genomics. So I'm very much looking forward to this session and learning along with you guys about all the amazing work that's going to be done by the faculty who are presenting today, and of course, their students. So our next speaker comes from the Medical Center, Professor Chao Lu, who is a professor in genetics and development, and he will talk to us about genetic dissection of epigenome partitioning. And I just want to emphasize that it's one of the really amazing things about this department is that we have a vast network of faculty, which you will hear from two of them this afternoon, in addition to the core faculty that you heard a lot from already today. All right.
Thank you for the introduction. Maybe I should take that. Yeah. Thanks for the introduction, and also organizer for the opportunity to share with you some of our work on genome packaging and partitioning, or we often call them the epigenome regulation. Our cells, your DNA in linear form extends over 2 meters, and our cells are very much I think about as your cell phone is that face two daunting challenges. One is to package this 2 meter long linear DNA into a tiny nucleus of about 10 micrometer in diameter. And also very much like your cell phone, you have this about 2 gigabytes of data where you have to extract information in a very dynamic and precise manner. Just think about the different apps on your iPhone where you need to track the right amount of data at runtime when you need it. So the cells obviously have come with evolved many mechanisms to deal with the genome packaging and access challenges, and it happens at the many levels. But we have been very interested in sort of the basic level, which is this structure called nucleosomes. So what this structure is, this crystal structure you show here is octamer of proteins known as histones, and it gets wrapped around about 147 base pair of DNA. So this is the basic structure of genome packaging, where you imagine the easiest thing to package something is to like wrap around your string on a series of beads, right, to reduce the volume. So the nucleosomes are not only important for the packaging, but it's also very important for the genome access. That is because all the histones have those kind of unstructured tails, and those tails actually have a lot of chemical modifications on those histone tails. We call them histone post-translation modifications or simply epigenetic modifications. And on top of that, the DNA itself can actually also undergo direct chemical modification. So, for example, 5-methylcytosine, the old DNA methylation in simple terms, is something I'm going to focus today. So all those combinations of those modifications sort of demarcate your genome and plays a very important role in partitioning your genome into different epigenomes, which correlates functions with whether your gene is active or silenced or poised to be expressed in very different states here. So obviously, for cells to add or remove those modifications, you need enzymes. So indeed, there are different categories of enzymes that are served to adding those modifications, remove them. We call them writer or erasers. There are enzymes that kind of read those modifications, so dock to the right side of the genome and carry on downstream function. So all those enzymes has been discovered in the past 20 years, and we know a lot about their function, both in development and diseases. But I want to highlight more recently that we have actually been using their function to engineer the epigenome. And the way to do is using the CRISPR-Cas9 tool, which we'll hear a lot more from the next speaker, Alberto. But simply by tethering the enzyme to the CRISPR-Cas9, and you can actually precisely locate this enzyme to a specific region of the genome. So you decorate local genomes with the modification you want to achieve gene silencing and gene activation effect here. So just to show you a serious paper on this one enzyme, which is the focus of my talk, DMT3A, which is the DNA methyltransferase, adding DNA methylation. So the paper shows it's not only possible, it can be scaled up to do screens, but most recently, actually, you can do that in vivo to achieve both efficient as well as durable gene silencing in long term. So that's pretty remarkable. But so far, we know a lot of those enzymes in terms of their business and the catalytic domain that actually putting out remove those functions. But enzymes are actually more than that. They actually have a lot of regulatory domains. Those domains are in generally poorly understood, but they're actually very important. So one of their functions is that they actually can sense what other epigenetic modifications are wrong in the genome. So the enzyme can naturally go to the right part of the genome to do their job, and also sort of catalyze their reaction in the right context here. So this is really forming a sort of communication channel for two different epigenome modifications to crosstalk with one another. And this has been very important for partitioning our epigenome into different domains, euchromatin, which is highly expressed genes located here, and a different kind of heterochromatin, which means the genes are generally silenced here to various degrees. And it's a combination of those different epigenome modifications and the crosstalk between them that actually set up the partitioning right here. And this is also generally what is known in development biology as the epigenetic landscape. So it ensures that once your cell undergo differentiation, it will stabilize in that differentiation state. So it doesn't just suddenly your brain cell turn to the skin cell out of nowhere, right? So it's a very important mechanism. So we have been very interested to understand the molecular basis of the regulation of those chromatin modifying enzymes, both in terms of their normal, since we think that's not only important to understand their normal roles in development disease, but also eventually will help us also better engineer those enzymes for things like epigenome editing. So today I will only focus on one line of the investigation in the lab, the DMT3A. As I mentioned, it's a DNA methyltransferase. It's already being used for epigenome editing. It's one of the two de novo DNA methyltransfer, means that it can take an unmodified cytosine and adding a methyl group on the 5 position here, 5-methylcytosine. And once it's added there, there's another enzyme, DMT3A, sort of propagate. So whenever cell divide, they can actually carry on this modification forever for some organism for millions of years. So it's a very faithful mechanism of demarcating your genome. So DMT3A is a major enzyme here, and it has this is the business end of DMT3A, which is the methyltransferase domain, is a catalytic domain. However, you can see there are several other domains here that are functioning very relatively less understood, and I really want to understand how the enzyme knows where to go, and once it's go there, what directs the enzyme to be active or inactive. And how does that contribute to diseases that are implicated by those enzymes here? So first question is, how do they go there? So we often do in the lab is just to map the different protein DNA interaction, but also the different epigenomic modifications using techniques such as chip sequencing. So you can actually, on the genome-wide scale, know where different things go. So this is a typical chip sequencing experiment where you can see here, this is our protein of interest, DMT3A, across the part of the genome. This is how their distribution pattern. And this is a related enzyme, DMT3B. But we are looking at two histone modification right here. So histone H3, lysine 36, trimethylation, dimethylation. So the exact name doesn't matter. There are two different modifications. But you notice that the DMT3A on the genome-wide level actually tracks quite well with the distribution of this one particular modification, H3K36 dimethylation. And then you can actually analyze on the genome-wide level where the higher you have on K36 dimethylation, which in those orange dot genomic regions, the higher level of DMT3A binding here. There seems to be a correlation. But is there a causation? So we can take cells and kind of delete the enzyme that deposit this histone modification, H3K36 dimethylation. So the enzyme name is NSD1. So as you can see here in the Y type of parental state, this modification occupies this very large intergenic region. So there's no genes here, but there's this vast intergenic region is well marked by this histone modification. And this is also where DMT3A is localized to. But upon the deletion of this enzyme, while the gene-associated K36 dimethylation remain unchanged, you can see there's a specific loss of this modification at intergenic regions. What's pretty cool is that the enzyme DMT3A now also lose their specific binding at those regions where genic region is not affected, and you actually see loss in DNA methylation compared to the Y-type situation. And we further studied the mechanism, and we found this particular domain, which is not essential for its catalysis, but nevertheless is important for its function because this domain actually specific binds to H3K36 dimethylation. So there's a clear mechanism by which the enzyme using this domain localized to this region and carry out its function over there. Okay, so summarize this part. So basically, we think that quite straightforward. The enzyme has a domain that reads a histone modification that allow enzymes to localize the intergenic region normally. But what if this domain is disabled, right? So we haven't really thought about it until actually we found a paper that reporting there's a patient that have a developmental syndrome have a mutation in this domain called PWP domain. So we decided to look at it. To our surprise, you would think that if you disable this PWP domain by mutation or just simply get rid of the domain, the enzyme should just fall off the genome and that goes nowhere. Instead, we found the enzyme is still attached to the genome, but just become redistributed. It still goes to a different part of the genome right now. So the Y-type enzyme sort of end up in this part of the genome, which correlates very nicely with catalytic demethylation. But when you get rid of the domain here, you can see there's a very profound redistribution that actually now we start to track with a different. So we were surprised to see, even if you delete the major way of how this enzyme gets to the genome, they're still there. So it really suggests that there's some other things that are going on here. And we did a little bit further digging, and we found that now the enzyme goes to a different place. This place is marked by a different set of histone modification. Again, the name doesn't matter, but this is the histone H2A lysine 119 ubiquitination. Right? So this is actually a pretty well-studied histone modification, and we're surprised. So we went to see whether does the enzyme actually interact with modification. So we can purify the enzyme, we can incubate them with the nucleosome carrying different modifications, and we see that indeed, if you look at the full-length enzyme, in addition to K36 demethylation, there's another interaction here with H2A K1 ubiquitination here that's missing in DMT3. And I'll try to go very quickly just so get out of speak at the time. So we then did a bunch of experiments. Not going into the detail, we found that what actually happened is a competition model. So the enzymes actually are getting dragged to different kind of the regional genomes through several different competitions. So when you lose one, there's another part of the enzyme which we believe is on the N-terminus through this serious experiment that actually mediate the enzyme goes to the H2A K1 and we found the region, and we actually solved the CREL-EM structure. You can actually visualize this molecular interaction using the microscope. And you can see very nicely there's an interaction of this particular region in the N-terminus of the enzyme. We named them, give them the name of the UDR domain, and here. And then we can actually, using that information to design mutation that make the enzyme lose its ability to go there. And again, we show here with introducing this one missense mutation or changing one amino acid enzyme completely loses the interaction to K1 ubiquitination and are unable to adding any demethylation at those regions here. So I will sort of end here science with the summary. So even this one little enzyme, there's actually a lot we still don't know about it. And the regulation is actually quite complex. What we think, what we know now, there's actually a competition how this enzyme goes. There's a sort of the canonical PWP domain interacts with the K3 demethylation. And also that's generally where this enzyme goes. So appears to be the winner under normal condition. We don't know why, we're still working on it. But even in the absence of this, there's another domain that makes the enzyme to a different set of regions. And we think in certain disease when this interaction is disabled, because humans do carry mutations here, and you can see this abnormal localization of DMD3A and abnormal methylation in some gene promoters. And we also think that by just understanding this, it would actually allow us to understand a bit better. Let's say if you use the CRISPR system to do epigenetic editing in different chromatin environment, you probably have to read the enzyme may function differently. That's something we need to consider in the future. With that, I'd just like to thank the lab and also the people who did work, the funding, and sorry about the hookup, and I'll end my talk here.
Thank you, Professor Liu. All right, so our next speaker is Professor Alberto Chicha, also from the same Department of Genetics and Development, and he will share with us about dissecting the DNA damage response network with CRISPR-based screens.
Thank you, Alan, and thank you to the organizers for inviting me. So in my laboratory, we're interested in studying the DNA damage response and various aspects of the DNA damage response. So how proteins that maintain genome stability regulate replication fork, remodeling, restart, and the repair of double-strand breaks. We're also interested in understanding the interplay between the DNA damage response and anti-tumor immunity. And more recently, we've been interested also in using functional genomics approaches for studying the DNA damage response, and in particular, for studying mutations of the DNA damage response, and for studying interactions between genes of the DNA damage response. So as you probably are aware, we are really constantly exposed to DNA damage, which can originate from exogenous sources, such as ionizing radiation, UV radiation, chemical agents. And there is also a lot of DNA damage which is induced endogenously in our cells. For example, upon reaction with the oxygen species, stress during DNA replication, spontaneous breaks, conflicts between transcription and DNA replication. And so the cell, in order to avoid and fix all this damage that it's exposed to, has evolved a signaling network known as the DNA damage response that maintains genome stability in response to DNA damage. And two components of this network are the ATM and ATR kinases, which are activated by sensor proteins, and they lead to the phosphorylation of hundreds of effectors with the help of transducers in order to promote cell cycle arrest, DNA repair, and when this is not possible, induce cell death or senescence. So as I mentioned, the DNA damage response is a highly complex network of physical and genetic interactions. It is enriched in many essential genes and large proteins with multifunctional domains. Most importantly, there are more than 40 genetic syndromes that are caused by mutations in various components of the DNA damage response. And there are tens of thousands of nucleotide variants in clinically relevant databases, such as ClinVar and TCGA, but the vast majority of these variants are of uncertain significance. So by studying these nucleotide variants, we can better understand the function of these complex genes and also determine the clinical relevance of variants of uncertain significance. So in order to do that, we utilize a system known as base editing, in particular cytosine base editors. This consists of a fusion of a Cas9 where one of the two catalytic domains has been inactivated, so only introduces NICs and not upstream breaks in the genome. And this Cas9, NICs Cas9, is fused to a cytidine deaminase, and it's targeted to specific locations in the genome using guide RNAs. And once it's targeted to these locations in the genome, it can induce the deamination of cytosine into uracil at specific locations in the genome. And normally, uracil is removed by uracil and glycosylase, and through base excision repair, you can then restore the original sequence. However, this fusion contains a uracil glycosylase inhibitor known as UGI, which prevents this pathway from occurring. And then the nicking activity of Cas9 can nick the strand opposite to where the uracil has been introduced. And following mismatch repair and DNA replication, you can then obtain a C to T transition at specific locations in the genome. And some time ago, we showed that using cytosine base editors, you can generate 32 different amino acid substitutions. And in particular, you can convert 4 codons, coding for 3 amino acids, glutamine, arginine, and tryptophan, uniquely into stop codons. And we call this approach iStop for induction of stop codons. And we show that iStop is particularly useful when you want to generate knockout clones without introducing double-strand breaks, because double-strand breaks can be potentially deleterious, given that they can lead to chromosomal rearrangements. And so in particular, for clinical applications, you want to avoid the formation of these rearrangements. And so approaches like iStop are particularly useful for disrupting genes. And recently, iStop has been utilized in the clinic, in particular for engineering T cells and disrupting the T cell receptor or various CD markers. And these cells were then further modified to introduce chimeric antigen receptors, recognizing CD7 on T cell leukemia cells. And these cells were then used for treating patients. And in a clinical trial in the UK, 3 patients were treated with these engineered cells, and 2 of them are currently still under remission, really indicating that these approaches for engineering T cells are particularly useful for clinical applications. So besides being interested in studying the functional mutations, we've also been interested in studying the entire set of mutations that can be introduced using base editors. And so to do that, we generated a library of 37,000 guide RNAs targeting the coding sequence of 86 different genes of the DNA damage response belonging to multiple DNA repair pathways. We then, using this library, we infected mammary epithelial cells, stably expressing a base editor, and then we either left the cells untreated or we treated them with various DNA damaging agents. We then selected we extracted the genomic DNA at the final and the initial time point and calculated the log-fold change in guide RNA abundance between these two time points. And this allowed us really to identify more than 2,000 guide RNAs that will show significant enrichment or depletion across conditions. We were able to plot the guide RNAs on the protein sequence of our proteins of interest, such as in this case 53BP1. And we could show on the top guide RNAs that were promoting resistance to DNA damage, or at the bottom, guide RNAs promoting sensitivity to DNA damage. And we can also infer the type of mutations that are generated by these guide RNAs and determine whether they are synonymous, missense, nonsense, or splice mutations. And this approach allowed us to identify new functional domain and interaction surfaces in proteins, allowed us to identify loss, gain, and separation of functional mutations, and also allowed us to classify variants of uncertain significance and determine whether they are likely pathogenic. So this approach relied on cell viability as a readout. However, recently, in collaboration with a group of Yadler Goblom in the Biological Sciences department here at Columbia, we utilize our base editing screens in combination with optical screens developed by the Goblom lab, known as CRISPR-MAP. So in these approaches, we perform base editing screens in breast cancer cells, and then we either treated the cells with DNA damaging agents or left them untreated. At this point, we didn't measure changes in the viability of the cells, but we fixed the cells and then imaged them to identify the sequence of the guide RNAs, and at the same time also determine changes in components of the DNA damage response and show whether certain factors were upregulated, were recruited to sets of DNA damage. And so we could link particular mutations to changes in the response to DNA damage by looking at these markers. So we are really excited about these approaches because we think they can allow us to define the functional nucleotide variants, to better understand the functional genes, because they can allow us to define disease-causing mutations and develop mutation-specific therapies for improved precision medicine. And because they can also allow us to identify the targets of small molecules and also mutations that induce drug resistance for improved drug design. So in addition to being interested in studying the functional mutations, we've also been interested in studying genetic interactions within the DNA damage response. And why this is important? Because studying genetic interactions, we can better understand the network of the DNA damage response. And we can also identify synthetic lethal or suppressor interactions that could be particularly useful for treating tumors and genetic syndromes with mutations in the DNA damage response. One of the classical examples of synthetic lethality is the synthetic lethal interaction between BRCA1 deficiency, which is very frequently observed in breast and ovarian cancer, and the inhibition of poly-ADP ribose polymerase 1 or 2. And this synthetic lethal interaction can be suppressed by disrupting a DNA damage response gene known as 53BP1. So we were looking at similar interactions like that, where we could see lethality when we disrupt the two genes simultaneously, and whether we could suppress this lethality by disruption on another gene. So in order to do that, we utilize a different Cas system known as Cas12a. And this system is particularly useful for combinatorial studies because it's able to process large arrays of guide RNAs that can then target Cas12a to different locations in the genome where it can disrupt multiple genes simultaneously. And so we generated arrays of guide RNAs where we could target either a single gene or two genes simultaneously. And in particular, we targeted 233 different genes of the DNA damage response, and we studied more than 27,000 gene-gene interactions in our screen. And we did a screen in mammary epithelial MCF10A cells, and in this case, we didn't treat the cells with any DNA damaging agents. We only left the cells untreated, and we were looking for changes in the viability of these cells in unperturbed conditions. So it took quite a while, but eventually the screen worked quite nicely, and we were able to identify negative or synthetic lethal interactions. These are interactions where guide RNAs that target two genes simultaneously give a phenotype observed, which is more severe than you would have expected based on the phenotype of guide RNAs targeting the two genes separately. And we found several known synthetic lethal interactions in our screen. We were also able to identify positive or buffering interactions. These are interactions where the observed phenotype for guide RNAs targeting two genes simultaneously is less severe than expected, based considering the phenotype of guide RNAs targeting the two genes separately. And also here we found several known interactions, including interaction between BRCA1 and 53BP1, which I just described. This allowed us to generate a large genetic interaction map where we are shown in purple synthetic lethal interactions and in green buffering interactions. We were also able to identify genes that show a similar behavior across the genetic interaction map, suggesting that they are functionally related. And oftentimes, genes that are functionally related also cluster together on the genetic interaction map, and they are also components of the same physical complex, such as the NCM8A9 and HROB helicase, or the components of RNase-H2, or the MUSD1-EME1 nuclease, or components of the Fanconi anemia pathway. We were able to validate this interaction using competition assays, where we are mixing together green label cells with the red label cells targeted with control guide RNAs, and the green label cells were treated with the guide RNAs targeting either the genes separately or in combination. And by doing microscopy, we were able to image and see whether there were changes in the growth of these cells compared to what we would have expected. And so through these studies, we were able to validate 27 out of 42 interactions, so more than 60%. So we're quite excited about this database. And so I told you in this part of the talk that these combinatorial CRISPR knockout screens allow the identification of interactions among genes, both positive and negative interactions. And they can be particularly useful for identifying dependencies in cells with deficiency in clinically relevant genes. And I didn't have time to tell you that we found synthetic lethal interactions between components of the Fanconi anemia pathway, which is frequently mutated in tumors, and genes that maintain genome stability during mitosis. And finally, I just want to thank people in the lab that conducted this work. The base editing studies were conducted by a former postdoc in the lab, Pierre Bion, and the base editing screens by another former postdoc in the lab, Raquel Paya-Martin, and the combinatorial knockout screens by former student in the lab, Sam Hayward, with two current postdocs, Alina Vitsenkova and Tomas Lamadias. And we had several collaborations at Columbia and South Columbia. And so thank you so much.
All right, thank you so much, Alberto. So let's go to our last presentation. This will be given by Professor Elham Azizi. She's anchoring the conference for us, and she will talk about how ML strategies can be applied to model the dynamic tumor environment.
All right, so we've had a full day of inspiring talks, and before we head over to the fun reception, I'd like to briefly just tell you about some of the exciting work in the lab at the intersection of machine learning and genomics in cancer biology. So we've heard a lot throughout the talks about why tumor heterogeneity is important. It plays a big role in, the cancer patients responding differently with this heterogeneity across patients, between the primary metastatic tumor patients as well as over time, and even within a tumor sample, you have a wide variety of tumor cells, immune cells that are infiltrating, support cells, endothelial cells, and so on. And so the big question in the past decade has been, can we leverage, a recent cutting-edge single cell technologies that have been introduced before to, in an unbiased way, dissect this heterogeneity, and single cell technologies, really took off and became scalable with these microfluidic technologies that encapsulate cells inside droplets together with barcodes that allow you to take measurements and at the single cell resolution. So in some of my previous postdoc work, we use this technology to understand what are the differences in the phenotype of immune cells in the tumor compared to the healthy tissue from the same patient. And we did this in breast cancer models with a suite of different computational tools. And one of the major insights was that we have this significant increase in the diversity of immune populations once they interface with the tumor. And a surprising finding was that the immune cells, for example, here shown are subsets of CD8 positive memory T cells, also showed different levels of expression of pathways and signatures related to responses to environmental stimuli. So, why are they showing these different and combinatorial expression levels brought us to this hypothesis that maybe this is because immune cells are sitting in different regions and locations inside the tumor. And so thankfully, with the advances of genomic technologies, we now can resolve the location of cells and find out if each cell population that has a different phenotype also has this link and dependency with this location. And the analogy used in the field is, before we were just tasting the average of fruits in a smoothie. Then with single cell, we were able to sort the fruits. With spatial, now we know that oranges are never sitting next to the strawberries. So that's the kind of difference that we were hoping to get with applying single sequencing-based spatial transcriptomics to. Here shown is a primary breast cancer tumor from a triple negative patient. This was a collaboration with Memorial Sloan Kettering, and we found quickly that in these complex tissues, it's actually hard to disentangle refined immune subsets, for example, activated immune cells from exhausted and immunosuppressive cell types. And also, previous tools, analytical tools, weren't really taking advantage of this histology imaging that the pathologist currently uses in the clinic for the diagnosis and annotation. They are also very limited methods to be able to compare the spatial organization of cells between different tissues. So 3 brilliant students in my lab, Siu, and Ashil, developed Starfish, which is an algorithm that combines the histology imaging with these high-dimensional spatial transcriptomic measurements to get this a joint embedding representation that can tell us, what are the neighborhoods of cells that have similar compositions of cell types. And just very quickly, what's the core idea and concept behind such a machine learning method is that we use autoencoders to map high-dimensional data sets to this joint low-dimensional representations. And they're called generative models because we put this in the framework that is probabilistic, so you can actually generate new samples from the distribution that you're learning with the decoder. Now, a lot of the work that we do in the lab is, adapting concepts in machine learning to address questions in biology. So if you just take, such an architecture applied on this data, these low-dimensional components are not going to be interpretable. There's still going to be mixtures of different immune cell types. So we really had to guide them with both either known markers of cell types, and in some cases where we have no idea what are the markers of major cell types in the tissue, we use this mathematical concept called archetypal analysis, which intuitively finds the extreme states that the cells measurements can reach that represent our refined cell types. So what are the insights we gained with Starfish? For example, in intratumoral heterogeneity, in the sample that I was showing before, the tumor cells that are located here at the bottom of the tissue compared to those sitting at the top of the region here, really have different phenotypes. They're more basal states in this region and more mesenchymal states, meaning prone to metastasis in the upper lobe. But interestingly, we can now start look at interactions with the immune cells, and we see that the co-localized cells with these basal tumor cells are more central memory T cells, for example, whereas here we have more effector memory T cells, and then on the margins of the tumor, we have more activated CD8 T cells, and similar observations in the myeloids. So indeed, our hypothesis was correct that different immune cell states are associated with different niches within the tumor microenvironment. Now, another question was, what are the differences between spatial organization of cell types across tissues? For example, in metaplastic breast cancer, you have, they actually grow on chemotherapy. They have a very different response compared to other triple negatives. They also have different morphology. So the question was, are the immune cells sitting, having different cellular wiring inside these tumors? So we collected data from metaplastic patients as well as other subtypes of breast cancer. Applying starfish, we always get this joint embedding shape like a starfish, so hence the name. And then we can define, what are the types of spatial hubs or cellular hubs that are shared or distinct to different cancer subtypes. Just highlighting one interesting finding, we found that the metaplastics, we have these concentrated pockets of immunosuppressive cell types, such as Tregs, regulatory T cells, and myeloid-derived suppressor cells that are overlapping in these hypoxic regions, also with tumor cells that are high in EMT signatures. We also validated this with spatial proteomics. So just wrapping up this part, with starfish, we were able to define different niches and cellular wiring that are specific to different subtypes of breast cancer. Now, the spatial dynamics is informative of cell interactions, but to really understand what pathways and transcriptional programs in a target cell are influenced by the neighborhood of that cell, where we have ongoing work led by Justin and Kushi that use attention-based mechanisms such as those in transformers like ChatGPT to not only use a position encoding and design in a smart way that tells you how close the cells have to be to really influence each other, but also get interpretation about gene-specific effects. Other work in the lab are, Bugino and Josh, who's also here, are working on making this multimodal to understand combining spatial metabolomics with transcriptomics. How is the metabolic processes that vary across the tissue, how are they influencing tumor immune interactions? Now, another perspective to look at cell-cell interactions is through temporal dynamics and how the cells co-evolve together over time. And a big focus of our lab has been understanding, immunotherapy, and this has been touched in the earlier sessions through Parisa's talk and Santi's talk. But our goal here is to, really use the power of AI to learn about the principles underlying immunotherapy response or resistance from instructive and successful cases of immunotherapies. In the case of leukemias, or liquid tumors, these successful cases have been CAR-Ts and also adoptive cell therapies, which were the basis of CAR-Ts. And a collaboration we had with Kathy Wu's lab at Dana-Farber, we looked specifically at an established type of adoptive cell therapy called donor lymphocyte infusion, and identified this interesting subset of T cells that is CD8 positive cytotoxic T cell, also high expression of this Hobbit or ZNF683 transcription factor, and it showed significant expansion in the responders in AML. So this was from our discovery cohort. We then took these markers, looked at a larger cohort of 53 patients. The results are reproducible. Our collaborators took these T cells and co-cultured them with the leukemia cells from the same patients to show that indeed they are reactive only to the leukemia cells, and they're also clonally expanded. So at this point we were like, oh, we found the needle in the haystack, and is this the magic ingredient that the patients need to respond to therapy? Are they directly interacting with the tumor cells, or do they need other players to coordinate the response? So for that, Cameron and Jovic developed a very cool method called DISCO. We're really proud of this cover illustration here under the DISCO ball, cells interacting. I'm not going through the details, but they found that it can model the temporal dynamics of these T cells that expand over time in the responders in the top row and not in the non-responders, but together with other immune cells that expand after them through a cascade, they inhibit the leukemia populations in the responders. What is really nice about DISCO, it tells you how interactions change over time. So before therapy, you have this high population of leukemia cells and some weak interactions shown with the edges here between the immune cells and the responders. But as the therapy is given, you see the significant expansion, but also stronger interactions between the immune cells coordinated by the cytotoxic T cell population. We validated this with protein measurements in the spatial level, showing co-localization and diverse immune hubs in the responders pre-therapy. They turn into expanded T cells with effector memory phenotypes post-therapy. In contrast, in the non-responders, we just have lower diversity of immune cells and T cells that are exhausted. We also showed this with spatial transcriptomic technologies, with actually technology that I just learned was developed in Professor Zhu's lab. Singular Genomics has now licensed it. So the take-home message of this story was, the bone marrow diversity as well as the phenotype of the T cells are really shaping response to immunotherapy, and similar to the cold and hot concepts in solid tumors. So I'm going to skip the next part of my talk because I'm out of time. But just to summarize, this is the big picture work in my lab. We develop tools, machine learning tools, combined with genomic technologies, profiling to understand questions about cellular wiring, determinants of response or resistance, and their underlying mechanisms that I didn't really get to talk about. Really grateful to the brilliant people in my lab who actually do this work, our collaborators, funding sources, and thank you all for your attention.
Thank you, Alham. So now I think we're open for questions for both Dr. Chachi and also Dr. Azizi. Sorry.
Yeah, my question is for Professor Azizi. I was wondering, when it comes to, congratulations on DISCO, by the way. It's a very exciting illustration and love the names. But I guess one of my questions was,
The students take the credit for the names, no.
it's great.
I'm bad at it.
They're catchy, right? So it's really great. But how do we account for maybe latent epigenetic factors when it comes to, assessing cell-to-cell interactions in mechanisms like DISCO? Because that might cause maybe a false overlap where it doesn't exist. So was just curious about that.
That's a great question. And it could definitely be an extension of DISCO to actually combine single-cell ATAC-seq. It's usually harder to do this in longitudinal samples from patients, but that can definitely be something that can be combined. We're doing this in other work, like multimodal integration of whole genome sequencing and single-cell RNA to put the transcriptional changes in the context of the genetics. But the epigenetics is definitely another layer that can be a great idea for integration with the ATAC-seq. Future work.
Okay.
Hi, excellent presentation as well. So you're looking at all these interactions in at the cellular level. And for a long time, we've been looking at similar phenotypes at the radiologic level. So we look at the microenvironment. We can see that there are different signatures from CT scans, MR scans, whatever, that predict response to immunotherapy, let's say, for non-small cell lung cancer, for triple-negative breast cancer. So we hypothesize that they might have something to do with microenvironment, TILs, things like that. We can't really prove it. And this is a study I've been dying to do for years, right? It's a difficult study because sometimes you may have to do prospective enrollment. You might have to find ways to establish a mapping within the scan and the underlying tissue before you excise it. Tough for the lung, but maybe doable for the breast. And then do all kinds of slice and dicing, multiplexing, all kinds of technologies there to try to establish this association. But if we could establish this association, then we could have a virtual, I don't know if I call it virtual, I don't like to call it virtual, it's real, it's just different real, right? Like an in silico, let's say, assay for tumor heterogeneity. That could be run routinely, clinically, and very inexpensively. Do you think something like that is feasible, or is it wishful thinking.
Absolutely. No, that's definitely our dream. So I think we're already learning a lot from even, limited samples about, for example, in other cases in colorectal cancer and not carcinoma. We were just looking at some xenium data. We're exactly seeing this observation that, even here we saw the proportions of this cytotoxic T cell population was enriched in, for example, responders post, but the other cell populations don't have a difference in proportion, but they do have a difference in the spatial organization, right? So we can identify these niches with spatial wiring that can be correlated with response, associated with response or resistance. Now, I think you're asking a more ambitious question of how can we now do simulations and counterfactuals, and I think moving towards foundation models, that's exactly a dream, longer term dream, to be able to simulate if, this patient was treated with this therapy or had this level of additional lymphocytes infused or inhibition of a specific subset of Tregs or exhausted T cells, would that make a difference in the response, right? And I think that's
Imaging phenotype. And the imaging phenotype of that response.
Yep, with integration of imaging, all data modalities, just like how we can integrate imaging, text, language models that are, to move towards foundation models with integration of different data modalities, that will definitely be the case in combining imaging. Yeah.
All right. I think one final question. Okay, I have a question for Dr. Chachi. So I think it was really powerful to see how you could screen the DNA damage, right? So is there any way to sort of leverage the CRISPR and potentially repair along the way, or have you thought of have you explored that already?
You mean to try to use study DNA damage response to improve the efficiency of CRISPR editing or repairing more DNA damage?
Or to repair the DNA damage that you have identified.
Yeah, I mean, could be possible, for example, to identify some interactions that will show increased repair of damage. It's something that would be interesting to see if there are some mutations that will increase the capacity of the cells to repair DNA and maybe make the cells more resistant to stresses. It's something that could be interesting to study. And also, from a therapeutic point of view, you can find mutations that will lead to resistance to drugs that maybe they do that by improving the ability of the cell to repair the damage induced by certain drugs. And so, these are all things that can be studied for sure. Yeah.
Exciting. Next year then. Yeah.
Thank
All
you.
right. Okay.
Yeah, so the methodology, there are very different ways of doing it, but it's basically you sort of take your genome and you sort of take your cells, you break it up, and you sort of release the nucleus. And then what you do is you use often antibody that are specifically recognize your protein or modification of interest, and they sort of bring down the DNA with it. And then you sort of use next generation sequencing to sequence what comes down with your antibody that suggests your protein of interest is binding to that piece of DNA. And then you map back to the genome, and you can see, depending on how many reads are actually coming from there, you get this kind of peak thing where higher peaks means that there's more enrichment of your protein beings at this particular region of the genome. Yeah.
That's a great question. So they don't have a lot of time to describe everything. So great. I'll go to the student and I will come back. Yeah.
Thank you. My question is about the earlier presentation, specifically regarding the in vivo testing, and this is probably for Elham and Jose. But so, for genetic testing, when we're looking to see interactions in vivo, one of the challenges can be that certain models maybe evolve faster, or changes in the models are very, the mouse models evolve faster. Maybe there's death because of GBM, etc. So I guess my question is, how do we study in vivo genetic interactions and take samples to study those when there's models that evolve very fast or change very fast?
So I guess is the question asking, just to reiterate, so this is looking at the interactions between cells, like
yes.
the T cell, GBM cell, But if we were to take it in the in vivo context.
Yeah, if we're sampling from in vivo, how do we kind of maintain the model so that, there's reasonable,
Right. Well, I think in the
standardization?
context of, I'll start that one when we're doing the genotype interaction screening, even in vitro. So that first, that middle story was done in vitro. We don't necessarily know. We know the types of phenotypes that are currently in GBM cells that induce programs that are induced by the T cells, but we don't know the one-to-one correspondence. You can imagine making some system, but this is something that would be amazing to figure out how to do some kind of tracing of very transient interactions in such a way that you can map one to the other. Doing it in the in vivo concept would be also very exciting. I think there are people working on that, so it's not. But yeah, I think that would be for us. Even in the simple in vitro system, we're not necessarily keeping track of that. I think in the there's definitely a lot of inference methods that Ellen is probably going to talk quite a bit about how you infer those tumor T cell interactions. From the screening side, when I make a particular genotype, then that's something that I think right now is challenging to be able to keep track of. But.
Great. I think there was another question there. Okay.
Okay. So I have a question about these methylation, the 5 positions, like for Dr. Lu. For example, the methylation is 5 positions, the carbon-carbon bond, extremely strong.
Yeah.
How many enzymes involved to remove this
Yeah.
methylation?
Good question. I have on my slide, but I skipped over it. There's a group enzyme called TET, T-E-T. There are 3 of them in mammalian genome that serve to remove this. The way it removes this is actually hydroxylate, and it has repetitive oxidation of this mark eventually removes it. But it's a very efficient process. So therefore, demethylation among everything is considered to be a very, very stable modification. There are active enzymes do it, but they do it in a very low efficient manner, and it's just in general, once you put it there, unless you really try to get rid of it, it's going to be there for a long time. Especially every time when cells divide, there's new DNA come, but there is an enzyme that can kind of recognize the old methylation and carry it over to newly synthesized DNA. So it's almost maintained through mitotic cell division. That's why it always has this memory of this modification even after many, many cell divisions.
The other about this methylation landscape, the function, right? For example, the promoter region, it's well known, but how about this gene body on the exon? If you have methylation, etc., what's really the function of those?
That's a fantastic question. The short answer is we don't know. That's what are we trying to find out. Clearly, their promoter we know much better, but there are many other regions that are methylated, and we can see the DMT3A goes to those regions. There's some hypothesis. None is sort of universally accepted. Some of the idea is that even at a gene body, as the transcription goes, you actually need some repression in the gene body to prevent aberrant splicing. So that's one function where there's some data to support that. There's some other ideas which that you have a lot of repetitive elements in your genome. And even in the gene body, you actually have a lot of viral genomes, ancient viral genomes that's invaded your genome and sort of stay there. And those genomes, even doing active transcription, you don't want those elements to be aberrantly expressed. So demethylation serves as a protective function at those regions there. Yeah, but I agree. It's an ongoing investigation. We're actually looking at that now in the lab, and we don't have a clear answer.
Okay, great. Those are good questions. Let's entertain a few more. Okay. Back there. This one. Yeah, thank you.
This question's for Dr. McFarlane Figueroa. One of the things you mentioned, I guess in the second portion of your presentation, was evaluating sort of the robust cell death and like processes that these cells undergo. Do you see any relationship between the S-associated protein and the proteins, or like, key regulators in this process with the cells you've worked with?
So is this on? Well, I can yell. So I can say that for the models that we use, especially for the first story, the patient-derived models, for the most part, they do have a lot of the genetic changes that I don't have to share.
Shout.
Oh, they don't have. They do have a lot of the genetic changes that are quite frequently found in the disease. All three of them have amplification of EGFR, so that's something that if we're looking for models that have EGFR dependence, then you want to go for those. And two of them have an overactivating mutation in EGFR. So, yeah, they do. They span part of of what we know in terms of GBM biology.
All right, great. Any other questions? This is your time. Okay. Okay, so I think I'm going to invite our chair up here, and we figure we can turn this into, while we're doing engineering back there, to turn this into an impromptu Q&A for the department. So see if you guys have any questions that you want to shoot at him. Not me. I just hold the microphone. Yeah.
And of course, we're obviously not electrical engineers. In Oregon, we do much better.
Yes.
But yeah, question.
Just thinking about how, like, many of us in this room are what is, like, one piece of advice that you or any other professors or grad students in the room have about, like, choosing a program and what you're looking for, or just one thing that you wish you had done in the process?
Oh, great question. I mean, I think one thing that is, actually, I just gave a little presentation in my lab to make it kind of clear, even to my students, like, what is a PhD and, like, why should you get it and why is it different than, like, other degrees, like bachelor's and masters. So this is my own personal opinion. So if anybody disagrees, it's fine. But, the key with a PhD, so when you finish your PhD, what you should be in the position to do is ask really good questions, scientific questions, right? And that's very different than kind of what you've been taught or essentially your education so far, because typically what you've been doing is solving other people's problems and questions, right? So here's a test, do this, and so when you think about the PhD program, it's a little bit uncomfortable at the beginning, or at least I think it should be, in the sense that you don't know what the question you're gonna answer, right? And in fact, you should, in some sense, be comfortable with being uncomfortable about that, because it's going to require a little bit of time. So as an example, I was just meeting with one of my PhD students. I don't know if he's here. Is Nicholas here? I don't want to put him on the spot, but so he's like a third-year student, and so we were meeting today, and he's like, Paul, I had this epiphany 2 weeks ago. I finally know the mountain I'm going to climb, right? Which was, he's been struggling a little bit about, like, what's the problem he's going to work on, and you guys are all very successful, and you're used to getting A pluses and things like that, and it's when you don't know what your problem is, what your thesis project's going to be, it can be uncomfortable, right? And some people find it sooner than others. Some people go into a lab where somebody says, you work on this. I would highly recommend you avoid, and this gets to your question, go to somewhere where you think you're going to have enough flexibility to ask good questions. Now, of course, a good question, and let me know if I'm just running on too long, because I could talk forever. A good question, a good scientific question is not like, so I do neuroengineering. Like, so how does the brain work? That's not a good scientific question. It's too general. And you have to think about the question within the context of, given the tools I have, right, as a researcher, or available to me in the lab that I'm in, and the area that I want to study and work on, will I be able to ask a question that allows me to generate hypotheses that I can test that lead to better questions, right? And so when you think about a program, think about it in terms of the flexibility that it allows you, and also really the principal investigator. Do they want to just have you, be a machine and do what they want to do? I would say part of your job is, if they do, but you really want to work with them, convince them you have other ideas. And quite honestly, I know when somebody's ready to graduate, when, you know, they have questions where I go, oh man, I wish I came up with that idea. So anyway, I don't know, it was a little convoluted, but happy to answer more.
I think just to add to that, I think what's really unique about the PhD experience, you know, as I tell my students, is that you have 4 or 5 years to fail, you know, within a cocoon, right? It's frustrating. It's going to be really crazy, but really builds character. No other way to put it, because I remember when I finished my PhD, I was like, man, I never want to see that project again. And I feel like I could do research in any area. Right? Because you did that once already. Then, you know, it's just maybe a little more need time to read up about brain-computer interfaces, you know, learn how to code, you know, there's no small feat there. And, but really you have that skill set, just like Paul said, to identify questions, to ask the right questions, and know how to iterate and go deeper into something. And I think that's really a really rare opportunity to train yourself to be a thinker, right? And what's really interesting that when we graduated with our PhD, there's really few things you could do. Now you could do so many things. Everybody's looking for someone with a PhD, really for your ability to think and to solve problems. All right, there was a hand there. Yeah.
I thought that was fantastic advice, so thank you for that. There's so many decisions, you know, upon us, but how might specifically, I'm talking about the decision, what do you do after you graduate? So how might your mentality change as you go through a PhD program, just depending on your desired trajectory after graduation? And when along your professional career did you realize you wanted to enter into academia / become a professor?
That's a great question, because it turns out I didn't really take a traditional path. So, you know, after I was finishing up my PhD, when I was done, I was a little bit, you know, I was burned out. I was like, and I wasn't sure I really wanted to go in academia. And I remember I started interviewing for industry jobs. And my advisor was like, no, Paul, you should be in academia. Go do a postdoc, do this and that. And I was like, no, I want to actually make a little money. I want to, like, you know, get out of the university environment. So I went and worked in industry for 6 years. During that time, I worked at a place called the Sarnoff Research Center. So we still did research, but it was a little bit different than academia in the sense that we had to, like, really think about the research topic, how it applied to a particular company that we maybe had a contract with. That was a time when Sarnoff was involved in spinning out companies, so I learned all about talking to VCs. I got really good at writing proposals. And so after about 6 years in the company, I was like a little burned out from there. I was like, all right, I'm going to try academia. But the thing that I didn't have at the time was I wasn't publishing lots of papers, right? And in fact, I would publish conference papers, but not journal papers, because I never did a postdoc. So I thought I came into the whole thing a little bit, let's say, disadvantaged from that perspective. But from the perspective of knowing how to sell a research idea and bring in money, I had a huge advantage, because postdocs never got there. So I came, you know, I interviewed at a bunch of places. I interviewed here. I think the thing that got me the job here is that the first chair, Van Mao, you know, he thought I looked like Harrison Ford. Now, I was much better looking back then, maybe, or whatever. So he'd introduced me. Didn't the guy look like Harrison Ford? So, but when I got the job here, I could, like, put a lab together. I knew how to manage people. I learned management at the company. And so I took this route that my advisor, my PhD advisor, was worried I would, you know, never get into academia if I didn't do a postdoc. And it turned out, I think it helped me. I think the other thing is, and I was a little bit surprised I didn't say 7 or 8 years after that, I'm ready to go, I don't know, farming or do something else. But being an academic and a professor, there's lots of great things, which include everything from, you know, not just the research you do, but, you know, you can consult on the side. You do a lot of, you spin out companies. I have 3 companies that have spun out from my lab. But then, every year, new young people come in, and they actually force you to think, and you learn from them. So, don't ever let anybody tell you have to do this, and this to follow your career path, and allow yourself to be adaptable too. It's tomorrow I might say, I don't want to be chair of this department anymore. And then I have Helen be chair. And she'll gladly do it.
He's dreaming. All right. Okay. Thank you so much, guys. And thank you so much to the team for the AI support. Okay, Chao, you ready to continue your story?
No, we are not ready yet.
Oh. They got me all psyched for that. No, just kidding. So actually I was going to, almost? Okay. All right. So I think, all right, I will have a few minutes to share that, I went the more traditional path. But I have to say, it wasn't because I really knew I wanted to go into academia. I knew I did not want to go to industry. So I did an internship in my junior year in Cambridge. Lived in a frat house. Don't ask. I will talk about that later. That at Siemens Medical Electronics in Danvers, Massachusetts. So they had a site there. So our chair at the time in bioengineering at Penn, Professor Thibault. So he actually, made these connections for all the juniors and said, hey, if you want to go for an internship. So I went for an internship. And, it was a great experience. I mean, Siemens, right? So you just sort of learn, what they were doing, and they were building different kind of devices. But I realized that they offered me a position to go back later at the end. And I was like, during the summer, I was pretty much done with my work about 3:00, So then I was like, oh, can I do more stuff? They're like, don't worry about it. Just go do your stuff. And I was like, okay. So when they offered me a position, I was like, oh, should I take it or not? And then my supervisor, to this day, I really thank him. He said, Helen, you know what I'm doing, in the evenings. I'm like, I don't know. And then he's like, I'm studying for my master's. So he said, you should go back, to school, get advanced education and think a little about, what you want to do, because you can come into companies at a much different level than when you are as a bachelor. So that was the advice I took. And, do I need to share more personal details? Or are we done there? Okay, awesome. To be continued.